problem is like if you sign up for all 4 classes at once, you get a discount on your 4th class.
Oh, do you really?
Oh, yeah.
But since I
didn't know that.
like sign up all at once, and they're like, yes, just do yourself.
That's
We
bogus.
should give you a break.
And we'll see. And I might be back. I think, doing fun things. Couple years ago, I think. Don't have to do other people's school.
What do we think the cookies in the middle are?
It's the landscape urban stuff. So there's also much in there. It's like the class size of
Oh, that's kind of like Icon, the one Chris and I went to visit. Break. It was pretty cool.
New York.
No, that Christmas break.
Oh,
That was in Idaho. I wanted
You
to
should
go.
cool to go the coolest places, and then there's me.
my cool place is like I
Yeah. I went to Butte. Nice.
Nice.
What kind do I think these are?
I would love to go to school.
Have
and
you
eat
opened the
toast.
raisin?
You think
Oh
so?
no, those are toffee.
Ooh, are they good?
Cookies.
Well, I don't know
Probably.
if I don't know if I like toffee. Do they not even give us
Wait, do you
chocolate
have a-
cookies?
Chocolate.
Chocolate?
What's that?
Oh yeah, we have.
Oh, my tattoo?
Yeah.
Oh,
Have you had that?
yeah. I know who Natalie noticed it today too. It was kind of funny.
I was like, oh my god, that's so cute.
Well,
Yeah, it probably has chocolate in there. It looks like, but Yeah.
You can give us.
toffee and chocolate.
And then lemon water.
It's so bad for the teeth.
Oh yeah, toffee is the worst.
Oh,
it will rip teeth apart.
What's this? Is this
pumpkin maybe? I don't know.
That's just the coffee.
Yeah.
all of them and try all of them?
Good point.
I don't know about the oatmeal raisin one.
Try it. Do it.
No.
I dare you. You dare.
Ooh, they have tea too.
No, like this one. I think I got 25 mostly.
This idea is fine.
That one I didn't know at all.
There's some confusing a little bit because it's in reverse from what we've done before,
Mhm.
which I was like, I know it's possible I said how it breaks apart.
Nothing too exciting, but
Like the intermediate approach.
Tea is really good though. Mint tea.
You know
Is
what?
that the one that you got was
Yeah.
mint?
Is that it's not like breaking it down?
I think I'm also
Usually it
mint.
breaks it down.
Oh, on shake, yeah, I said things about it.
No, it's really frustrating.
How have you been?
How's pretty good. Really good.
I'm like, I need to.
What class are you touching right now?
well, I got botany
Which one was it?
Oh, nice.
Black 5, and the first week we were able to do some field work even, so that was nice.
Oh,
Yeah.
that's cool.
Yeah,
Usually it's probably too cold.
yeah. Well, you know, the first week we went out 3 days and did some really cool field work,
Oh, nice.
and you know, we had other stuff to do inside then,
Mhm.
computer members and what have you.
And I bet the class wasn't as cool as our class.
Are they?
I bet that class wasn't as cool as our class.
Probably not. No, I don't think it was.
That
It was
class
good,
was
though.
fun.
It was a lot better than I thought it would be because it worked out to do some really cool things out, which I'm gonna
Did you get your
always
results
try
back?
to do.
Oh, yeah, we're good.
Yeah, just be
awesome.
flexible,
8 more months and I get another whole body CT
For
scan,
sure.
and we're good.
you know, because of the weather, I'll have to be
Mhm.
flexible with it, but I'm hoping to always do that. But yeah, your class was really cool.
Yeah, I had a lot of fun. And with the
Good.
rattlesnake too.
I know, 2 rattlesnakes.
Yeah, that was crazy.
Rattlesnakes.
That was crazy. I know what you're talking about. That was funny.
Is it just the way that I said it? No, when we were in botany, we were up at the M and we were doing field work and like measuring out plots of specific types of plants that we
Did your
picked.
body fit what?
No, I did it first.
No.
So it was this year, but we were walking down and like it was like on the, I don't know if is that the north side or the south?
Well, it's a wide valley,
Mm-hmm.
And so it was right on the bottom of a wide valley,
That's
and it was getting the sun.
Yeah.
So they were
just
hanging
soaking
out,
up the sun.
soaking up the sun.
Well, and there were two of them.
Really?
So
Mm-hmm. As
The
soon as
one-
we saw one and reacted to that and, looked at it
never seen
and went around it, there was another one.
Oh wow.
Never seen
They were
that.
big, too.
9,200 feet.
No way.
Dead serious, I have a video of it.
That's
It's,
crazy.
in the backyards, they're on fire, the Mount Baldy fire in 2020. And we were walking, I was like, took my dugout, I saw one. What's that noise? It was like, we were tucked under a rock. We're like, what are they doing up here?
Do they just give you random cookies?
Yeah, I guess today we got random cookies. We didn't even get chocolate. I was so bummed.
I thought that was standard.
That's what I was thinking. But I grabbed like 13 chocolate cookies, so. I don't know, she got I grabbed 3, so.
How's it going?
Oh, it's good. Yeah. I like science.
So where do you think you're gonna
It's fine.
apply in Utah?
So I already applied to grad school
It's just
and
Keeps you
applied
busy.
to 3 grad schools. Got accepted to one.
Oh, that's so exciting.
accepted to
Yeah,
one, wait,
like
wait,
understanding
wait,
it
wait,
kind
wait,
of.
But the one I got accepted to, I don't want to.
Anything new?
Which one is it?
It's
You teaching this class?
it's like CRAHN up in
Oh,
New Jersey.
I think it's going well.
Which
It's like
one is this?
cramming and cramming.
Issues in biology.
Oh, okay. Is this the syllabus that I just saw?
Yes.
Oh, wow, outstanding.
Oh.
And do you have, a lot of gen eds and majors?
Oh.
What's the distribution like?
I only have one major.
And how many gen eds?
11?
That's great, they need that.
Oh,
Yeah,
that's
they-
exciting.
I don't know why
I
I
would
didn't
want to go
fill.
somewhere on the East Coast. I wish I would
12
Yeah,
have.
I'm
students
surprised.
School while I was
I'm
there.
surprised as a gen ed that it didn't go.
But it's still listed as an experimental course. But
Yeah.
it should be on the regular schedule. It's like 102, I think, or 103.
Right, that would have been
Look
outstanding.
who it is!
Hey,
Man of the hour.
nice to see so many of you here.
Well, I'm super
I told him.
We did human evolution today. Tomorrow we're doing cooperation, conflict, and behavioral
Our
ecology.
keynote
talking
speaker of the
about child
day.
marriage.
Hello.
How are you, Steve?
Yeah,
Fitness benefits.
So
How's it go?
Evolutionary
yeah, it's
psychology.
pretty good. You could ask them, they're in the class.
Should be interesting.
Oh,
Yeah,
what they're,
learn
and
this.
Yeah,
that one, I ask them all the
wanted to
time.
hear his opinion.
They're doing terrible.
Are they?
Yeah, I think it's going pretty good. It's hard because we had to jump right into things, right? So,
Kids are more involved.
Get up to speed.
Tough crowd.
Can't forget OChem 1.
but yeah, OChem 2, I think we have to do preschool labs. So we get to do like,
Who's in the class?
so we isolate
Kyle.
ibuprofen from a tablet.
Talk to
and
him
we
every
try
day.
synthesize
He's
aspirin
so
from
weird.
salicylic
He's
acid.
awesome.
And then we make banana oil. We're doing that one.
Is your wife
You know,
protected
Acid
the
smells like
animals?
banana.
Yeah. What is it called?
She's still on
Isopentyl acetate.
Oh, I know.
Okay. Because that's what contaminates beer, right?
I
Is it a
went yesterday
byproduct
and
of
I was looking
fermentation?
for her. I want to meet her.
Acetate
Acetate-
compounds. Yeah,
acetate
acetate
compounds.
compounds definitely give
I think
flavor
it's
to
a
beer.
banana.
She does
But I think it's
exist.
I just
also,
don't
know.
believe it
it
though.
was, it's
Does exist.
unfavorable.
I would think if you had,
Yeah,
probably you
sure.
don't want your beer to taste like, but smell like bananas, right?
Yeah.
I always thought that some beers like you want little fruity like side
Sure,
notes.
sure.
I guess it depends on the beer.
I went to
Maybe
a
it's lager.
fermentation class one time
You know what I'm saying?
at Weight Labs in San Diego.
I don't know.
Oh,
If she's,
okay,
oh, I
yeah.
know she's blonde hair. That's what I've been.
They,
told. So
they
she's
talked about it, and I
blonde.
hadn't heard of
I've
it.
never
Yeah.
seen
I'm
her
pretty
hair.
sure it was some acetate. I think it was
She was not
bananas.
Do you make beer in one of your classes?
I don't, but that's something that might be fun to do someday.
We had like I took a yeast
Art
genetics
science.
class
My goal.
that just only did, we just made beer as part of it, and then we did all the
Faculty's
isolation
private
and then
lives,
genotyping
be
and stuff
private.
like that.
Everyone's met her.
But one thing we could do is you could bring in
It's a true
like
statement.
unfiltered beer and
Have
you
you
bring
guys
in and
ever
try to
heard? Oh, I
isolate
think
the yeast,
you
you know,
should.
so we
Chris?
put like chimay
Are
and
these
try to
raisins?
isolate the yeast from
Yeah, I
that.
think they're.
I was gonna say, they look
Yeah.
very raisin.
Yeah, cool.
No, she just saw.
No, that would be fun though to do.
That
I've always thought it'd be great.
I've never even
Yeah.
I'm certainly not an expert, but at that
made eye
time,
contact.
that was
We do the same.
fun to learn more.
TV or?
Because they're one of the main companies that sell the yeast.
Yeah, I think I've seen that
Did you
for a
hear
little
some
while. I
of
got
the
into
talking
Duane and I would buy from White
No,
House.
I didn't too.
Packets we had like the yeast.
I did yell.
In a liquid form, as soon as you add it, as opposed to like dried yeast, it takes longer to get the fermentation going.
Yeah.
Yeah, that was kind of cool.
We talked
It was a
about
great,
these,
a great 2-day
Neither-
conference.
none of
Because you
those.
got to drink beer the whole time.
Yeah, that's what they do. Right?
What did you learn about
I was
invasive?
reading about some, where did I go? Like,
What'd you learn about
we were in
invasive
Bozeman, there was a brewery there, and they had
Yeah,
a
you
whole bunch of
philosopher,
different beers
you
we got.
Yeah.
That was,
I was,
it was
yeah.
made with engineered yeast, that genetically engineered to produce different flavor compounds. So it had like a really kind of
Yeah, that
unique
is a
profile
much
to it.
higher
But I think a lot of people were
rated
hesitant because they
product.
saw genetic engineering, you know, but
But hey, when there are
beer makers do
ecosystem
claim that this is the
effects,
future of beer.
We
You
call
like
it biology.
tailor the
Yes.
genes that you want to add to get whatever flavor
We do.
profile you want as opposed to having.
when there are
some sort of
ecosystem
selective
effects,
yeast, right?
Oh, that's cool. I saw something.
leaders
but
are
I
getting kicked out, they're booted
I'll
out,
look it up. Because now I'm intrigued.
and you're starting to wonder
Something to do with
if
horses.
our
But I
native
think
consumers
remember
are
there was a
are
yeast
having
that could
trouble
produce
because of that.
Evolution and
You're
action.
having these negative feedback loops
Actual
associated
whatever
with
acid.
this invader.
That is
I mean, what else are you gonna
Yes.
talk
When I- when I-
about?
Just to look it up.
When I was reading
Yeah.
So you
about
wouldn't
it,
have,
So
to-
you wouldn't have to
hop.
you could just have the right yeast in there.
Right.
And then maybe like, sometimes it's uncontrolled, you have all the other compounds that come out the hops that maybe you won't want, so you can just get the
You didn't even
one.
comment on my ceiling,
Right?
I'll have to look it up. I can't remember if that's, I don't know if that's right.
about you
Yeah,
being
that's
an
kinda
invasive
cool.
species.
Yeah.
So I'm introduced.
We're in
Oh, cool.
Really?
I just wanted to know if
You're talking about.
In Louisiana.
Yeah, I mean,
I haven't seen it
you
yet.
guys that were here on the
I
first
know,
talk,
I know.
I've seen a third of the slides, and they're the same.
That's good.
But I'll get a little bit more into this.
And did you see the,
Information.
I put it through, did
Audience,
you see that?
And way more back.
I had to go.
Information.
Tell Maddie.
Kind of tailored it
Not to
exactly
spell
on that.
scenario, but she wrote
Okay.
sequoia, which is Spanish
What year
for
was it?
assassin.
December of 2000. Yeah.
Right
You're
before
like,
COVID.
where did you get
Yeah, I'm
assassin?
glad I got out of there.
Did you read our?
Yeah, yeah.
Spring.
It was like.
Our
In December, when the
paper
first
you
cases in
graded the
China
fall
went on.
Yeah, and I think it's, I did a little postdoc for like 4 months to finish
Tell
up.
me all about how
That was
you
really
grew
down
up.
there. And it was right, I feel like it was only a month after.
Bracken and I were
They
key
were gonna
graded
shut down everything a
Brecken
little bit except
wasn't
essential
as
research
focused
activities
as
because
it
they
should
were the
have
first
been.
people.
I learned a lot
You know, this is
about,
gonna be like the black.
hot shots.
Right,
Yeah, we didn't
No
know.
one
I was
knew.
looking for
Yeah,
the
and then it
ethics
was
for
like
your
they
paper.
had to work.
I could have,
with
I would
protocols
have
and everything.
Yeah.
For a while, it was like you had to be constantly tested every single day. You had to have only working on shifts.
When you were
and all
still
that.
there?
No, that was right.
Right after you left. Good time.
Yeah, if that had been right when I had.
I felt like I
the last
wasn't
of my
as
project.
clear on my expectations.
Yeah.
So,
Your grade was like
Clearing that
more
up.
by the late.
yeah, I guess that's true. Yeah.
Think about the
Clearing that
first.
up.
If you had to stress that, and not only you was like, I don't think I can do this.
Yeah,
but also
for all of a
it's
sudden,
like,
that,
I
oh my
think it's more
God.
difficult.
There's probably a lot of nutrition.
I rebuilt them from scratch.
I should have taken physiology this year, that's all I have to say.
Why?
You already said it's gonna be like so much more.
Hannah may help make it better for the fall.
Oh, is she gonna,
I don't know.
oh,
Let's see.
well, we'll convince her.
Wait, what is he gonna do?
I asked. I don't know if she's.
Is your online picture still shows how people land, right? If we Google you, which I haven't done.
Yeah, for research gauge.
When
Okay, when I
is
first,
the,
since I was on sabbatical,
12:30?
I Googled you. and
What's the meeting for?
like,
I thought
okay,
it was
he likes
a
that, he or she likes
Oh, it's supposed to
surveys.
be, but I didn't.
I like
email
to
it
stay,
her, so.
I'm trying to stay anonymous.
No one told us except.
But the foundation asked me for a picture.
I think sometimes
Why? I didn't
people that
respond.
push my picture.
Yeah, I actually,
Well, they just came to my office.
Exactly. They take your
I
picture
mentioned that you,
for
I sent out a
whatever,
massive
and all
email.
of those pictures and, online. I haven't
Speaking of which, also,
got it. They're there though.
I just sent out an email on Tuesday to Bio Club or something, and I was thinking, I should put on.
We're happy, yeah.
I'm
Yeah,
just-
sorry,
I'm
they're-
Yeah.
I did say that right now. But yeah, if I had had it my way.
I really should have got
Picture of planet, happily planted something.
Erica, did you talk to the woman who's
I-
looking for an expert in tribal artifacts?
I think that
Oh, why would I?
like 3 is fine.
They were going around
We're not-
building.
I almost said 3 is enough.
faculty,
The reason I'm.
who would be, I said, maybe,
No,
you know,
3 is
ask
fine.
Mom.
Treat yourself at 4.
Three pills, though.
As in her
I talked to
anthropologist.
the other
just for
person
fun.
going around the building, Girl Scout cookies.
This is how we, or actually.
We already
the rest I
signed
go
up,
through and just make
Saturday.
things, so it makes more sense to me. It's been awesome.
I missed that.
Yeah.
It's over here, though.
Very good.
What is it? Is it the call number?
No, that one is amazing.
She has a really cool attitude.
Ooh, what is it?
It's called Billy.
Ooh, what is it?
Mostly, I think,
You can like sync the slides to it.
You're gonna have to accept
I could have colored during bioethics, but we weren't allowed to have our iPads, so I couldn't stay.
It's an analog class. That's why.
Could have just colored the entire time. I don't ever do that.
You just rearrange basically class notes on a just.
Kinda, yeah, but I make it more so I can understand it and yeah, more happy friendly.
Oh, okay, so it's just all good.
Reactions,
I actually started doing that
it makes it so much better.
because I changed this because it shows up. It's
Yeah.
Just buy an iPad. It's totally worth it. My dad got me an iPad for Christmas and I was like, I'm never gonna use this, but I use it every day.
Really?
Yeah.
Thanks for.
I don't have any wanting to try to encourage people.
What did you ask?
We're just learning. Maybe I can convince my dad, I mean, give me this one.
Yeah, I told my parents to buy me an iPad, they told me that I didn't need right now, that my name was for fine.
You buy an iPad.
And my laptop's broke too, so I need to buy a laptop. Yeah.
It's $1,000.
What is? Yeah. My laptop was, I think, 1,200 and it lasted me 2 years of crash.
So that was my idea,
Oh, I was
So
really
it is,
talking about some of the,
this here.
the benefits of
What are the things like these? They're so good. And you get to like,
Yeah.
you get to anything else.
Look where it goes.
The grocery store, what are you doing?
Like, today's just like, there's no work, and then like, go back to the. My list of questions I have, you just start like listing all of them, check, how many those comments.
Yeah, no, I made a list.
Did you tell all your students to come speak?
yeah,
They get a little extra credit?
I don't give any extra credit.
Wow.
See how many are truly dedicated. That's right.
Then you can offer them
I know,
extra credit
it's
the next time.
Yeah, baby.
Your next one, you get one every week. We got here.
Oh, yeah.
Crazy.
I know, right?
Yeah, I don't know how many more topics I can come up with here.
Those ones are pretty ill.
No.
Thought about doing something crazy like on my phone, so I was like, no, it's too weird, it's too out there.
Do you want
That could
another?
be a really cool one.
It could be a cool one, yeah.
Yeah.
So
Uh-huh.
You want
it never
to?
gets done.
That I can.
Yeah, it'd be like a book tour.
Yeah,
Can
it
you
could,
bring
yeah.
copies of
So
this?
like, why don't I
for us?
I thought
I feel like I'm ready to do that myself.
Like one of my good friends, definitely, Michelle's husband, Jeff, he's almost,
Oh, that's cool.
got a signed copy of that.
Oh, yeah. That's good. Got a good one, right?
No,
No, Eric.
all the tall.
Yeah.
Short.
It's not poetry.
I just took this off.
If you had all these different patterns to follow, like iambic pentameter or something like that, like haiku, you have to memorize like 5, 3, something like that, right? So it was almost like, I'd rather just write something that had a bunch of knowledge, but you don't have to like worry about that. framework, we're getting everything provided. Something like that.
Those are the red violet.
Yeah, and then really, yeah, exactly, really good poetry that actually conveys something super deep by the end and has interesting sort of within stuff. There's something really cool about it.
Yeah, for sure. We need to put the language into original expression. That's cool.
My sister.
Did your wife come back yet?
Yeah, I know, she come back sometime this afternoon. She got back.
Yeah, I was like,
She might have come except for 2 little ones.
I feel like I've seen you around.
I'll have to look at the second floor. There's a daycare.
On the second floor?
Yeah.
I did see a couple of them. We just like, we don't know until they come and do that. So I got her some, I brought her one of the molecular sets.
Oh, did you?
She was really into that.
You gotta take a walk.
My mom.
Oh.
I don't know how many other schools in daycare
Are saying, where are my
molecules?
molecules?
For my molecules. I try to be like, okay, this is water, this is called ethanol.
There's a graphic
I
designer.
gotta say oxygen.
Oh, that's good.
So, yeah, it's pretty
Okay, we
good.
can give it
Yeah,
to.
Yeah.
It's awesome.
KGs and that. It's hard to do Yeah.
Yeah, I just kind of said if I can.
They might have a
friends, 'cause
project
that's-
or we can maybe utilize some of those skills as well for this.
Yeah.
Oh, yeah, you can kind of see sometimes what's gonna pop in, like in the slides here. Usually I forget.
Yeah,
like,
right.
where-
They asked me.
You know, that's you get at yourself, it's like, oh crap, where was it?
Yeah,
Where was it? Where was it?
And I'll be like, oh, it's like, all right, and the next topic is blah, blah, I'll switch the slides like, oh, well.
Oh, I forgot
actually,
that.
Hold it, I forgot.
Yeah, are we?
You're the man, Steve.
Timeline would be
Good man.
trying to organize this, try to have a meeting.
Yeah, you'll be able to talk about CWD.
Are you doing some stuff where you think
Yeah,
talking about?
yeah, that's- yeah. We're at the assays up with that, so. Yeah, I should share that. Assays.
Yeah, that gives
Yeah,
us
I'd like
plenty
to
of.
I was working, I kind of haven't, never quite finished it up, because I was got distracted with this and started class. But eventually there was a couple of interesting suggestions.
Sorry, what?
The different ones
Heads
that
up.
we could run, and I kind of had a flow chart about which ones, you know, there's like, how you're gonna stage, which one first would be the most range, and then where you have more specific ones, and so maybe it's input on specific ones.
Yeah.
Yeah.
It's a big problem. You gotta find protein.
Yeah, I think there's,
It's probably not.
yeah, exactly, it's gonna be pretty low.
And also,
I found some methods that
that wasn't advertised.
That's like they had some.
I mean,
That's what they want you to do.
Just same thing.
and not
In terms of
I'm
time.
not sure with that. I don't know how that would work. You have to go through.
They always kind
yeah, probably. The first day.
of, and that's also why it's nice to
You
do.
can, but then, you know.
Put them in the bag here.
Right, yeah, I don't know. But it could be. I mean, and
Yeah.
there's positives.
It's kind of weird, they research that, because what they did, and it's, they're all are, and I think that doesn't seem to be an issue. There's like the potential.
Yeah, right.
To humans. It has to be, it's never, it's original. But if they've done laboratory studies where they cells, humans have they don't, well, no, they definitely see a change in cells. But then when you look at any sort of study, for people, they show no change. Stuff that's shown later. Probably smart.
Yeah. Especially because we're doing short. We were hoping to hire for.
Some people have a natural aversion to eating other parts of their
We probably should be going here.
Yes.
Oh, nice, yeah, I'm just gonna reach out to this girl.
All right, everybody, I think we're going to go ahead and get started. So thank you all for coming to our next installment of Life Inside and Out. Thank you, Bradley. We approved it, yeah. But we're honored today to have one of our own here speaking, Dr. Steve Perry, who's teaching chemistry, as most of you know, and are currently in class with at the moment. So Steve is going to give us a talk today on some of the research that he did while in graduate school working on his PhD at the University of California, Santa Cruz. Dr. Perry gave a talk not too long ago and talked about fatty acids and inflammation and all kinds of things. And so some of this same aspects of biology and chemistry, for that matter, will be talked about here in this talk. So, all right, Steve, I think that's ready for you to take it away.
Awesome.
Thank you very much for giving our talk.
So where's the light switch for the?
Oh, I'll grab it. It's up here.
Up there, yeah. You can see the screen a little better. Well, thanks for coming, everybody. Hopefully you can learn a little bit of something from this. If any of you guys came to the last talk I did, it'll be some overlap with the material here. So hopefully that just means you'll learn it really good. You'll ask really good questions now because you'll have seen the material a little bit. So like Mike said, this is a talk about the PhD research I did. And I've kind of divided up into 3 different sections. This is just one different project I did. But the first third is going to be kind of about just inflammation in general and a kind of different way to view inflammation than what we're usually used to. And so we'll get kind of heavy into that, into inflammation, and try to understand that a little bit. And then the second part is going to be understanding an enzymatic pathway that we're interested in that affects inflammation. And then the last little part will be like a little bit of some structural biology, which is where we're going to be looking at an enzyme, doing some mutagenesis on it and such like that. So we're going to kind of go from big and get really, really narrowed down on the end there. So we'll mix in a little bit of some biology with some chemistry and hopefully get a broad perspective on this. So to begin with, we got to understand how inflammation works here. And most of us know that, like, if you get some sort of cut, you get a splinter, general something like that, you get a process of inflammation that occurs in response to that injury to your body. So here you have a splinter entering the body, and you're going to cause some release of chemicals that are going to lead to the effects that we consider inflammation. So you get like redness, you get swelling and all. So one of the first things that happens is we get fluid diffusing into that site. We get white blood cells coming in. If there's any bacteria, we can fight that infection. And so this is a healthy, normal process that your body needs to go through in order to fight infection, in order to prevent outside organisms from getting a hold there and causing more damage, right? So this is a normal process that we want to have, but sometimes it can get out of hand. So we want to understand how that happens. And when inflammation occurs, the first step is fluid coming in. And the second step is some of the blood cells that are circulating around, specifically white blood cells coming into that area. So you probably heard about red blood cells, there's platelets, but we want to focus a little bit on some different types of white blood cells that are involved in inflammation. So there's basophils and eosinophils that are generally involved in allergic reactions. And lymphocytes, monocytes are kind of a more broader term. Lymphocytes produce antibodies like B cells and such. And here's neutrophils and macrophages are kind of what we want to focus on. So neutrophils are kind of like the infantry of the immune system. They first come onto the scene of inflammation, and they're looking to do battle with any sort of bacteria or anything there. They have a lot of aggressive digestive enzymes that they can release in order to damage any bacteria, help clean up the mess. And macrophages come, and they're called what's called a phagocytic cell. So it's PHAG, and they eat other organisms. So they'll eat bacteria, they'll eat other things, they'll clean up the mess afterward. So we're going to kind of focus in the research that I did on neutrophils and macrophages and some of their roles in inflammation. So we usually think of inflammation as being a process that occurs in response to an injury, and then it kind of just goes away when the injury goes away. But there's another phase of inflammation that we want to understand. So in the beginning, you have edema, which is like when fluid gets in, you get a bunch of swelling and redness. Then you have this influx of these polymorphonuclear cells here, which are basically just neutrophils. And then you have macrophages at the end there. So there's sort of a cycle of inflammation that happens. And in the beginning, we start off with really heavy, like fighting inflammation. And at the end, we have something called the resolution of inflammation. So we're kind of ending inflammation at the end there. And it's not just an anti-inflammatory effect. There's a difference between something that's anti-inflammatory and pro-resolution. And we'll see that's going to become important later on. So if we don't have good resolution of inflammation, then we end up getting chronic inflammation. So normally we'd have some sort of injury, we get an acute inflammation phase, where we're fighting infection, kind of cleaning up the mess. If that doesn't end and it goes on for too long, we end up getting chronic inflammation. So you get fibrosis, so you get like things, extra tissue deposited there that's not supposed to be there. You can get damage to nearby tissues. If you have like heart disease or something, you can get atherosclerosis, which is the buildup of plaques and such in your arteries. So we want to avoid long-term chronic inflammation if possible. And the way your body does that is generally through this process called resolution, right? So we want to make sure that there's an active end to inflammation and not just sort of a passive decrease in the ongoing inflammation. So let's just make sure we understand these differences here a little bit between something that's anti-inflammatory and something that's pro-resolving. So if we think of anti-inflammatory, we have a drug or something that's just preventing the ongoing inflammatory process, right? So that's these neutrophils coming in to fight infection. If it's anti-inflammatory, it's just telling neutrophils don't come in anymore. You don't need to fight the infection. We're kind of done. Pro-resolving is actually the cleanup that happens after an inflammatory event occurs, right? So if you take an anti-inflammatory drug, but you don't have good resolution, you can still be left with the lingering effects of inflammation because you're not cleaning up. And I kind of liken this to anti-inflammatory is just like a ceasefire during a war, and the resolution is actually the repair and the reconstruction that goes on afterwards. So we need to rebuild the tissues. Macrophages will come in and clean up all the neutrophils and sort of the debris of bacteria that have been destroyed, sort of extra bits of cells and such. So that active process needs to happen in order for cells to not go into a chronic state, right? Now, if we go back to this picture here, we looked at the different cell types. And it turns out a lot of what mediates what cell types are present during inflammation is a whole series of molecules that are called oxylipins. So they're lipid-based molecules that have oxygen in them. And in the beginning of inflammation, we get some molecules called prostaglandins and leukotrienes. And in the resolution phase, we get some molecules that have really nice names like resolvins and protectins and merisins here. So the switch between these two types of molecules helps regulate whether we're in an inflammatory state or a resolution state in the end there. And so basically, we look at the presence of these lipid mediators. We'll see a big spike in the beginning of these lipoxins, these prostaglandins. These are the pro-inflammatory mediators. And over time, in an area of injury, you'll see eventually that these specialized pro-resolving mediators will increase as well there. So it's really important that those molecules are produced in the right amount and that we have enough of them in order to prevent chronic inflammation from occurring, right? So I mentioned that they're made from lipids. So how do we get these molecules and where do they come from? So to understand that, we have to go a little bit into like lipid chemistry, lipid biology, and understand where lipids are in our body and how we get them and what happens to them. So fatty acids are primarily where we get these mediators from. So there's a few different types of fatty acids that we have in our body. So a fat is just a long non-polar molecule. It's kind of greasy, it's not water soluble, and it has just a couple atoms on the head group here on the end that help it go into water. So there's three different main types here that we have. There's unsaturated fats, which we usually think of as oils at room temperature, and they're kind of bent like this. That bent shape prevents them from packing closely together, so they stay liquid. And we have saturated fats like this. Saturated fats are what's usually in like butter or animal fat. It tends to be solid at room temperature. And we have these what are called trans fats, which are sort of artificially made. If we take one of these and we chemically alter it to make it straight, we get a trans fat. Those are usually viewed as not being particularly healthy. And we should touch just a little bit on how to name these because we're going to be talking about a couple different types of fats. So we want to make sure we understand how these unsaturated fats are named. We're going to be focused on these different types of unsaturated fat. So we have a terminology that's based around this omega name. So if we looked at a fatty acid, and this is a little picture of it, we've got our oxygens down on the end. These are all carbons. And from this end, if we're going to number in, we're going to count to the first position of a double bond here. We're going to call this end the omega end and this end the alpha end over here. So we don't have to worry about alpha. We're just going to look at omega because we're going to study some different fats and how these different fats interact with enzymes in the body, right? So if we count in 3 spaces before we hit the first fatty acid, our first double bond, we'd consider this an omega-3 fatty acid. If you came in 6 positions, that's an omega-6. If you had to come in 9, that'd be an omega-9. And we're mainly just going to focus on omega-6 and omega-3 because they make the kind of signaling molecules that we're concerned with here. So let's go back and look at fats, and we'll see how these kind of fit in. So we said that there's saturated fats. Here's an example, this long chain there. Meat, dairy, eggs tend to have a lot of saturated fats in them. And here's a monounsaturated fat. So that's going to be an omega-9 generally. It just has 1 double bond. That's where the mono comes from. And that's going to be in things that we usually think of as healthy, like olive oil, avocado, and nuts and such. And then you have these polyunsaturated fats. So poly meaning we have multiple double bonds in them. And there's kind of 2 different types of polyunsaturated fats. That's the omega-6 and the omega-3. And the thing is that there's a really big difference in the health of these, right? We wouldn't usually think of like corn oil and soy oil as being super healthy. Usually corn oil is not generally considered to be healthy versus fish, seafood, algae, generally considered to be more healthy, right? And we'll get into this a little bit more and see why. So we're going to focus on omega-3s and omega-6s and understand how we can affect inflammation by changing our diet a little bit back and forth between omega-3s and omega-6s, and basically what happens in the body when we do that. So let's look at a couple different types of molecules here. These are omega-3 fats here, right? So we've got this group in the front that makes it dissolve in water. We've got a long tail, and we can see there's a lot of different types of tails that we can get. Just for example, so in your diet, you might have this alpha-linolenic acid, which is 18 carbons long and has 3 double bonds. Or we could have something that's 20 carbons long and has 5 double bonds. Basically, we just have a lot of variability in the type of fat we can have. We also have a bunch of different types of omega-6 fatty acids. That's 6 carbons in from the end here. So the research that I'm doing is focusing on 2 specific types here, and we're going to kind of compare the effects of them. So we just want to be familiar with arachidonic acid, which we're going to abbreviate as AA, and docosahexaenoic acid, which is abbreviated as DHA. And the naming there, like this one just comes from naming docosahexaenoic, so it's got 6 double bonds and it's 22 carbons long. So anyway, DHA and AA are the 2 terms that we want to know here. And how do we get these? These are primarily going to be made or primarily gotten from our diet and not synthesized in our body de novo from scratch. So like omega-6 fatty acids, like arachidonic acid, going to come a lot of times from fried food. There's a lot of corn oil, soy oil in the food, in the oil that we're frying things with. Also palm oil, butter, coconut oil tend to be very high in arachidonic acid. Also chicken and meat, beef that's fed corn. Now, omega-3s tend to come from mostly cold water fish, so like salmon and tuna, mackerel, things like that. But also grass-fed beef tends to have a lot more omega-3s in it than beef that isn't fed grass. And also we'll see it in flaxseed and in chia seeds and walnuts have very high amounts of omega-3s. So again, just what we eat can really affect what fatty acids we're getting, because our body isn't super good at synthesizing that. So if we look at what our body does when we get these, say here's a couple omega-6 fatty acids that we could have in our diet, linoleic acid and arachidonic acid. And here's a couple of these omega-3s. And it turns out your body can convert between these. So your body can turn this omega-6 into that one, and it can turn this omega-3 into EPA and DHA, but it can't really make it can't go back and forth between these here, right? So if you have a diet filled with omega-6s, you're kind of stuck with that. If you have a diet full of omega-3s, you're kind of stuck with these ones a little bit. And ultimately, your body kind of synthesizes a lot of AA from whatever else it has, and it synthesizes a lot of DHA from whatever else you have there, but you're going to kind of be stuck with whatever type you get. So what happens with arachidonic acid and docosahexaenoic acid is that these react with enzymes called lipoxygenases and cyclooxygenases to make pro-inflammatory or pro-resolving mediators here. So the omega-6 fatty acids will react with the same enzyme through the same pathways, but the products will be mainly pro-inflammatory. And omega-3s will react primarily to make pro-resolving molecules, right? So how is it that these enzymes can react to produce those molecules? So let's just review a little bit about cell membranes and where we can get these fatty acids from. So if we all remember, all our cells have a little fatty acid lipid membrane around it, sometimes called the plasma membrane. And those are made up of phospholipids. So it's a little lipid and a phosphate head group. And so here's a picture of the membrane. It's got a whole bunch of fats in it. And if one of those fats were to come out here, and we're going to look at it up close, we can cleave it off with an enzyme called phospholipase. So say if your body wants to signal, it can take some of these fats, can cleave off an arachidonic acid that was sort of stored in the membrane like that. So then this arachidonic acid will be floating around. If it encounters one of these two enzymes, then it can make something, some sort of product here. And in this case, with arachidonic acid, whether or not we encounter a lipoxygenase or a cyclooxygenase, we're just going to get pro-inflammatory products, right? So interestingly, if we take some anti-inflammatory drugs, they're going to act on different parts of this pathway. So this action of phospholipase to make arachidonic acid is blocked by like cortisol and steroidal anti-inflammatory drugs here. So like if you've ever had to take prednisone or something like that, one of the effects is to block that. And if you take aspirin or ibuprofen, one of the effects is to block cyclooxygenase, right? So we have some good drugs out there for different steps in this pathway, but we're lacking in drugs to deal with the lipoxygenase pathway. So there's some interest in studying that. So one more slide just to kind of put this together and see what happens when we have different diets and how it affects this process. Is let's imagine a membrane here, and we can have either arachidonic acid or some omega-3 fatty acids in there, right? So let's say that you have a membrane that looks like this. It's got a lot of red sort of omega-6 fatty acids. If you eat a diet that's say high in fatty fish and walnuts, we can enrich our membrane in omega-3 fatty acids, right? So all of these fats in our membrane, these phospholipid tails, have way more omega-3s, right? So let's say something happens, you get some inflammatory event, say like a heart attack, which is technically called a myocardial infarction here. What happens is you'll release fats from the membrane. And if you happen to have a diet high in omega-6s, you're just going to release a ton of these omega-6 fatty acids. Versus omega-3s, you're going to have a lot more of that. And then just through the same pathway, the cyclooxygenase and lipoxygenase and other enzymes will convert these ones into pro-inflammatory mediators, and these will be pro-resolution mediators, right? So the exact same pathway here is just going to be pro-inflammatory or anti-inflammatory based, or excuse me, pro-resolution based, simply based on the mixture of fatty acids in your diet, right? So traditionally, the Western style diet tends to be very high in omega-6s, whereas some countries that tend to have lower inflammatory diseases, like for instance, in Japan, East Asia, they tend to eat a lot of fish and less corn and wheat comparatively, right? Or less beef, traditionally at least. A lot of places are adopting the Western style diet for a variety of reasons. So some of this is changing, but the traditional view was that. Okay, so that gives us a little idea about the background of inflammation. So how do we prevent inflammation then? So the traditional approach has been using some sort of medication. For instance, here's the white willow tree. And for many years, it's been a source of salicylic acid. And there's a few other plants that have been used around the world that have salicylic acid in it. And so this was a traditional medicine that was used for a long time. No one really know why it worked, but you could make salves out of it or make teas out of it.
it turns out this has a compound in it called salicylic acid. And in the late 1890s, this guy, Felix Hoffman, was working for Bayer Pharmaceuticals, and he was one of the first people to make a pharmaceutical. So he came up with a way that you could react this alcohol group here and acetylate it here. So that's the kind of reaction he did, is called an acetylation. And he invented aspirin here, which is acetyl salicylic acid, right? So this was the first sort of medication that was created through chemistry and not just natural means here. And it wasn't until much later that people figured out how this worked, that aspirin was working on an enzyme called cyclooxygenase. So this is a kind of space-filling model of what cyclooxygenase looks like. And cyclooxygenase reacts with arachidonic acid, that omega-6. And it produces these molecules called prostaglandins. And here's kind of a model to see how that works. Cyclooxygenase takes the fat and it reacts with the two oxygen molecules that kind of make this cyclic structure. Now, when you take aspirin, aspirin inhibits cyclooxygenase and prevents cyclooxygenase from making prostaglandins. So then you don't get the same inflammatory response when you take aspirin, right? But again, from what we learned, this is only half of the process, right? Because we've prevented the ongoing inflammation, but we haven't promoted resolution, right? So if you have you have a heart attack or something like that, this can prevent it from getting worse, but we wanted to find some way that we could promote resolution a little bit more. And part of that was understanding the lipoxygenase pathway. So lipoxygenase is another enzyme that's involved in making these mediators. So the research that we did in my lab was focusing on a set of molecules that were made by lipoxygenase here. So this is called a myrcene. It's made from DHA, so docosahexaenoic acid. And the difference is that we've just added two oxygen molecules that have then become reduced to alcohol groups there. But we're basically just adding some oxygen, and it creates sort of a signaling code, right? So where we've added oxygen on a given molecule can signal different things to the immune system. So we studied the lipoxygenase enzyme to understand how we could make these and how we could potentially inhibit them or modulate how much was produced in different circumstances. So let's talk a little bit about the lipoxygenase enzyme here and how that works. So similar to cyclooxygenase, here's a space-filling model. This shows that outside of a protein, a protein's just a bunch of amino acids that are folded up into a long chain here on the outside. And if we look inside to see what that looks like, we can see there's this long chain. This is about almost 700 amino acids long, and it's folded up into different shapes. We've got an alpha helix or beta sheets here that all wrap around. And in the middle here, we've got a little iron atom, which is the catalytic center of the enzyme. So we're going to kind of zoom in on that and just see what's going on with the catalysis here, right? So this is a little cavity inside the enzyme. If we think of an enzyme as being like a little Pac-Man, there's a spot where different substrates can come in and react. So here we've got an iron atom, and here we've got arachidonic acid inside this little cavity here, right? So let's see how this process works with lipoxygenase. It'll come in a little bit handy later on. So here's arachidonic acid. We're just going to focus on a couple of these little sections of double bonds here. So we've got 1, 2, 3 double bonds just in this middle section here and the iron atom. So when you react it with oxygen, you kind of have almost like a rusting of the iron atom here. It changes its oxidation state. And through that process, it's able to remove a hydrogen and add some oxygen here and change the position of the double bonds. And so that's how we add oxygen and then make this from a fatty acid into a signaling molecule, which can signal the immune system a little bit. So the key here that we use for our research is that we've gone from 2 double bonds that are separated, that are far apart like this, to 2 double bonds that are close together. We call that conjugated. And so this means that those double bonds can share electrons. So we get a little bit of a effect from having shared electrons versus non-shared electrons here. And so we use that to our advantage. We can see the way the enzyme operates using ultraviolet light. And so most of you guys will probably remember at some point in chemistry seeing this kind of image here of the electromagnetic spectrum, where we have radio waves that have a really long wavelength of like a mile down to microwaves and light, ultraviolet. So ultraviolet is right here. It's small enough that it generally interacts with atoms. So we can use a device called a spectrophotometer. We can shine light through a prism and then through a little quartz tube that has our reaction in it. And as we form those conjugated double bonds, we can observe the reaction progressing. And so we use this to measure the formation of our products. So the other thing we want to think about is that there's a couple different lipoxygenases, and we can add oxygen to a few different spots on a molecule. So here's arachidonic acid. We could react it with the 5-lipoxygenase, and it would add an oxygen to the 5th carbon, 1, 2, 3, 4, 5, of arachidonic acid. And so we just label this as 5-heat. We could also, there's a 12-lipoxygenase. It adds an oxygen to the 12th carbon. We have a 15-lipoxygenase that adds an oxygen to the 15th carbon. So we want to be aware of that. And then one slightly confusing thing is if we look at DHA, DHA is 2 carbons longer than arachidonic acid. And so if you react it with the 5-LOX, it adds an oxygen to the 7th position. So it's like slightly weird to think of that, but the DHA is just a little bit longer. So the numbering was made for arachidonic acid. So for 12, it becomes 14, and for 15, it would become 17. It's the numbering. So we're going to be dealing with some of these molecules here, and we want to make sure we understand that. So again, our big concern was looking at figuring out how this molecule is made, right? So we can already look at it and see we've got an oxygen at the 7th position and the 14th position here. So that would sort of imply a method that we could make this. If we remember, the 5-lipoxygenase could add something to the 7th position, the 14th could add it to, or the 12-lipoxygenase could add it to the 14th position. So we were curious how we could get to this route here. There's 2 different ways. Could do 5-lipoxygenase first and then 12-lipoxygenase, or vice versa. We just want to get a 14 and a 7 position oxygen here. Now this seems like kind of esoteric, but it's kind of relevant because different types of cells have different types of lipoxygenases. So macrophages have 5-LOX, platelets have 12-LOX. So depending on which sort of cells you have in a given area of inflammation, you can make different products. And the order of whether those cells come into a area of inflammation can then determine whether or not you can make something, right? Because it would be kind of odd for maybe macrophages to be in an area first and then platelets to come in later on. You'd think if you had a cut, platelets would be there first. So also there's some other problems here is if we want to make this product, it implies that we need the 12-lipoxygenase. But if you think of inflammation, a lot of the chronic inflammation that people have doesn't involve some sort of cut or a wound, right? You can have joint problems, you could have a ischemic stroke. And if we look at just a chart from an article in Nature, we can see that there's all kinds of different ways to get inflammation here. Like if just disturbing your sleep can cause increased inflammation, depression can cause inflammation, diabetes, you can get viruses. These are all processes that don't generally involve clotting, right? So we thought there was maybe something wrong with this because we couldn't figure out in all these conditions how you could get some of the inflammation you saw without having platelets around, if that was the method that had occurred. So another hint that we had is we developed an inhibitor in our lab that was an inhibitor for 12-lipoxygenase, so it should turn off 12-lipoxygenase. But when we gave it to mixtures of cells, we still saw this product getting made. So there had to be another route that this product was getting made by here. So we had also observed a little bit of a strange behavior in the 15-lipoxygenase, that most of the other lipoxygenases are very specific in what they make as a product. But we found that the 15-lipoxygenase, which should react with DHA to make the 17 product, makes a little bit of the 14 product, something like 10%, a little bit on the minor side there. So we thought, well, maybe the 15-lipoxygenase could be producing this product in the end there. It wasn't something that people had considered, but as we'll see in the end, it has important implications for what kind of cells are involved. So we'll look at this and add in the possibility of the 15-lipoxygenase making this product. So we started off and we were really curious, well, can the 15-lipoxygenase even interact with this? Can it react with it at all? So we did a kinetic study using our UV-Vis spectrometer. So we look at different concentrations of the substrate and we see basically how quickly the enzyme can react with different concentrations of it. And we showed, if we look at some data here, looking at DHA versus the 7-DHA product, that it reacts very similarly to it. So it's quite happy reacting with this oxygenated version there, right? Which is kind of interesting. A lot of enzymes are very, very specific for their product. So if this one reacts the same as the other one, that's a good sign there. So the next thing we wanted to look at is we wanted to see what products it made when it did that. So we knew it reacted, but we don't know where exactly it added the oxygen. So we wanted to use a method called high performance liquid chromatography. Some of you guys who have taken OChem before, we've done chromatography with a little column. This is the same, but it's really high pressure. So say you have some mixture of products, you react an enzyme with something and it produces a whole bunch of different things and you want to separate those out. We have a column with some packed material and it interacts with this mixture, different components of the mixture in different ways. So if we pump the mixture through here, this red material interacts very little with the column material and goes through quickly. The yellow stuff interacts strongly and is sort of held back as the fluid is pumped through here, right? So we get a separation of the components and we get a series of peaks coming out the HPLC like this. Each peak represents a different component that we've separated out here, right? And we couple that with what's called mass spectrometry. And mass spectrometry is an interesting technique that you can use to identify an unknown compound. So here we have this molecule here. And the way mass spectrometry works is we shoot it into this giant box here and we hit it with electrons, which causes the molecule to sort of blow apart into little fragments. So we've got all these different fragments here. So we can then measure the size of all those different fragments and make a plot here. We have all the different sizes. So here's a fragment at 341, 297, and 199. So this is almost like a little fingerprint. The pattern of fragments here can tell us what molecule we've gotten here. So if we have an unknown mixture, we can run it through this device and we'll have a really good idea of what we got. So we ran our products of the 15 LOX reaction through here and we got two main peaks out. And here's our mass spectrometry that we did. We can show the molecule and we figured out where all the fragments are and how it breaks apart. And we got a really interesting pattern that we saw that the 15 lipoxygenase has totally switched the way it's worked. And when it interacts with this molecule here, it becomes a 12 lipoxygenase. So it changes its activity. So instead of making the 17 product here, so we started with a 7 substrate. We should be making a 17 product. But instead we made mostly the 14 product here. So we call that it's switched its positional specificity, which is kind of interesting. And again, we'll show why that's really important later on. And it's kind of interesting enzymatically. Usually things have a certain product they made. They generally stick with that. They don't suddenly make a different product completely, later on with a similar molecule, right? So one other interesting thing that we did to study this was I showed this version of the molecule. But it turns out that you can get different stereochemistry, right? And if some of you guys have taken organic chemistry, you may remember stereochemistry is just sort of how these different oxygens are aligned in space. So the way this is drawn here, it's going into the page. This one's coming out of the page, but we could reverse those. We could change the way the double bonds are. So we could get a couple different versions of the molecule here. In this model, both of these ones are coming out of the page. And in here, we have sort of a different arrangement of double bonds. So minor differences, but another way to help us understand which one of these molecules were made, they have different pro-resolving properties. So we ran another technique that's called a chiral HPLC. So that's the high performance liquid chromatography. But by chiral, we can tell the stereochemistry of a molecule. We can tell whether it's what we call a right-handed or a left-handed molecule by running it through a special kind of column. So we were able to run a whole bunch of different standards through and determine that the molecule we made was distinctive here and lining up with this particular product as opposed to our other ones, right? So we can correctly identify which one of those three I showed on the last slide was the molecule that we were interested in. All right, so now we can correctly identify how quickly the enzyme is reacting and also exactly what products it's made. So what we wanted to do next was figure out, sort of combine these two together. And so we use this technique, it's called biosynthetic flux. And so we're basically just combining together a few different measures. So let's look at this and see if this makes any sense to us. Here's our 5 lipoxygenase, and here's what it's reacting with. And we're just saying it's reacting this quickly. This is just a measure of how fast the enzyme reacts. And it makes only this product, right? So our flux is going to be basically the same number here. Now, if we look at something different, if we look at the 15 lipoxygenase reacting with a different product, here's the speed, but it makes only 86% of the product, right? So we're just adjusting this final amount. What we're saying is the flow through this biosynthetic pathway. And so we're going to use that to compare them all. So what we did is we're going from DHA. We want to figure out how to make this. And we looked at all the different possible ways that we could do this. So there's several different enzymatic routes to make this intermediate and different ways to do that. So we've got all these possible options, right? So we looked at the kinetic, the rate, how fast the enzyme is, and what the products are for all of these. And then we're just going to adjust the arrows based on that biosynthetic flux to see how quickly they react, right? So this gives us a good idea of which one of these pathways is more likely to occur, right? So we've shown that there's a giant bottleneck right here. This is about 100-fold slower than this one, which is about 100-fold slower than the others. So this is going to create a huge bottleneck here. So we showed that we think that this is the most likely route for synthesis here, and also for another reason that we'll talk about on the next slide and why that's important, right? So this is using the 15-lipoxygenase, that unexpected new activity that we showed. And so this turns out to be kind of interesting for the transcellular biosynthesis, this idea that you need two different cells coming together with products here. Because previously it was thought that you needed macrophages and platelets to come together, and that doesn't work in all different kinds of inflammatory conditions. So, but it turns out that macrophages express both the 5-LOX and the 15-LOX at different points in time. So the idea that we proposed then was that macrophages can come in the beginning of an infection and just express the 5-lipoxygenase to make pro-inflammatory mediators. And then later on, when it switches to resolution, they will express the 15-lipoxygenase, which will then turn these molecules, these pro-inflammatory molecules, into, pro-resolution molecules as well. And so you don't need any other cell types, and this is just mediated solely through macrophages. And interestingly, there are two types of macrophages that we see in inflammation. There's M1 type macrophages and M2. So you have a monocyte, which is sort of like a baby macrophage comes into an area of infection, and then it differentiates into one of these two cells, right? And part of what we're proposing is that we did propose is that the M1 macrophages just upregulate one type of lipoxygenase and make the anti-inflammatory or the pro-inflammatory mediator. And just by switching its expression, we can make these pro-resolution ones in there. And again, also part of this is dependent on whether or not you have arachidonic acid or whether you have DHA primarily in your cells. So if you have a lot of arachidonic acid, then there's not going to be much for these M2 macrophages to do. But if you have lots of DHA around because you're eating lots of salmon or something, then there'll be tons and tons of DHA for these for the macrophages to make into pro-resolving compounds. All right. So the last couple of slides I want to talk about is really getting heavy into the structural biology here a little bit. So we were kind of curious how exactly the lipoxygenase enzyme can kind of switch its activity like this. And so we did some studies on different enzymes, different related lipoxygenases to try to identify what parts of this molecule's active site were important, right? So we identified a couple amino acids that were important here. So remember this is showing a cavity here inside the enzyme, and the outside of the cavity is defined by the different amino acids that live all around it. So here's one here. This is an arginine, and we've got a couple different ones here, leucine, and a few different molecules that we found that were likely to be important from comparing different lipoxygenases here. So we ended up looking at some of these and narrowing it down to a couple different ones and thinking that the main factor that determined how the arachidonic acid fit in the active site was these amino acids at the end. So we tried changing those amino acids and then reacting that mutant enzyme with the amino acids to see what would happen here. So here's just a little model of the active site. Here we're just drawing in those same amino acids that I showed before. Here's the iron atom. This is where the catalysis happens, where the oxygen comes in. And here's our arachidonic acid sitting in here. So here's the carbon 12 and the carbon 15. So they have to be aligned in a really certain spot in order for the iron to work and react with that. So in order, here's the same active site, but we're going to put the 5-HEAT or the 7-HDHA in it already. So this is the molecule that was going to be used to making the later doubly oxygenated compounds. And we're just thinking that this has to be aligned differently in the active site. So we said that if we started with arachidonic acid, we get the 5 product, or if we start with DHA, we get the 7 product. If we start with 5-HEAT or the 7-HGA, we get a slightly different product. So in order for that to work, the molecule has to be deep in the active site. So we wanted to look, we focused in on this amino acid here after looking at several other ones and not finding much effect. So this is a phenylalanine, and that number just tells us what number on the chain of amino acid works. So we're going to try to change this amino acid and see how that affected what the enzyme was doing. So here's a picture of that active site and showing where this is right here. So we could change this amino acid and then look at the percentage of different products that's made here. So the wild type enzyme, which is the normal enzyme found in cells, if we react it with arachidonic acid, it gets mainly the 15 product. Whereas if we react it with 5-HEAT, we get mainly this 12 product. So it's showing that it sort of shifted its positional specificity. So what we're hoping to do is make a mutant that can reverse this effect or recapitulate the other effect here. So we kind of look at some modeling here. If we change this big phenylalanine into a little tiny amino acid here, we've created a whole lot more space in the bottom pocket of the enzyme. So that allows this arachidonic acid to go deeper inside, hopefully allowing it to change its shape. So we showed if we make a more space in the active site, we should predict that it can go deeper in. And we see that we have shifted the product specificity here. So we're proposing that this depth of the active site is primarily what regulates that shift in specificity. And if we do the opposite effect, if we turn this into a tryptophan, which is a much bigger amino acid, so it jams up the bottom of the active site and prevents this to go in, we should see the opposite effect, right? Which is what we saw here. So we're getting 96% of the 15 products. So we sort of shifted this over. And the same thing here, we've shifted the product back towards what you should expect there. So that was kind of an interesting discovery there that we hope explains much of why we had this shift in with the 15 lipoxygenase. All right. Well, thank you all for listening. Hope you learned something. This is my lab that I was in at UC Santa Cruz. So my PI, really great guy, Ted Holman here, and just a lot of interesting people. I worked with a lot of really good undergrads and a lot of different grad students there, a lot of fun, a lot of interesting people. And we had some collaborators at the University of California in San Francisco and the University of Michigan. So to thank all of them and all of you guys for coming to watch. Hopefully you learned something. If you have any questions, please let me know.
Were all those undergrads purifying lipogenases for you?
They were doing a lot of purification, yeah, and a lot of mass spec. Yeah.
Then you threw them some mutants too.
Yeah, yeah. I feel like you can only run kinetics so many times before it gets a little boring. But so they had a few weeks of running kinetics before they started to realize that it was boring.
Wow, that's boring.
Yeah, it worked out well. They worked really hard and they learned a lot.
This is kind of a random practical application, I guess. We were just at this wilderness first aid training over the weekend and they told us it was no longer about like rest, ice, elevate, like you still do that kind of stuff if you have a sprain, but they were saying back off the anti-inflammatories to allow that process to continue. But of course, no context for what was continuing or why. So is part of that because of something like this where you have something like a macrophage come in and do inflammation, but also be part of the resolution process after?
Yeah, and a lot of it is because there's a balance here, right? Like you need that inflammatory step in the beginning. So say if you got rid of all the omega-6s from your diet forever, you would have problems, right? You would not be able to mount an appropriate inflammatory response. So much of the problem with, chronic inflammation and injuries is not having the correct balance here of the inflammation and the resolution, right? And I think part of that is, because of the cells that are present and some of the diet there. There's some interesting things that they showed. If you take really large amounts of omega-3 supplements, you can actually, you can decrease the clotting and your incidence of like heart disease and such, but you can also decrease your clotting. So you're more likely to suffer some sorts of diseases that, like I think a hemorrhagic stroke, or you're more likely to have longer bleeding times. I don't know whether it's clinically relevant, but they've shown that as well. So having the right mix of fatty acids in your diet is one way they can modulate it as well. I don't know if that answered all your question.
Yeah.
There's another part, yeah.
So with certain internal injuries that cause inflammation, where you don't have an external factor coming in, potentially causing like an infection, where you just have like joint issues, would the inflammation still be necessary since it's not necessarily fighting an infection? Are there other reasons why we'd want it to prolong? Or if it's just an internal injury, would we want to just stop it immediately?
Yeah, a lot of times the internal injuries, it's less important to have an inflammatory response. And so we sometimes mount a longer inflammatory response than necessary for internal injuries. A lot of chronic stuff that you might encounter, like rheumatoid arthritis or something, involves a longer process than is necessary because there's not really an injury, there's not an infection. It's really like this failure for the body to sort of clean up or reduce the inflammation in the area. And then it becomes like a self-propagating cycle of positive feedback, right? Where more and more cells come in. So we want to cut that process off and then help that body heal by upregulating maybe some of the enzymes involved in resolution, or perhaps changing our diet. You know, we were hoping in some of the research in this lab that we could find like say an activator that could, speed up certain enzymes, or that you could find that if you downregulated one of the lipoxygenases and upregulated another one, that you could sort of shift the balance towards pro-resolution at certain times.
So earlier, you were looking at some of the causes of inflammation, and you mentioned stuff such as disrupted sleep, other stressors. I want to say there's anything like the basically psychological stressor, I guess you could consider them. And I know that like stress has been found to lower immune defenses in the body. I mean, that's how kind of ulcers are able to happen. And I was wondering if maybe if some of those stressors could be causing inflammation due to the body kind of overreacting to a lower immune defenses. That was kind of panicky in a sense.
Yeah, there's been some interesting research lately, especially related to depression. And it's not totally clear whether or not depression leads to inflammation or whether inflammation leads to depression. But it does seem to be clear that there's some sort of connection between those two. And maybe there's some sort of feedback there. So there's some receptors in the immune system, like CB1 and CB2 receptors that interact with endocannabinoids. So there's some theory that it might be related to that. There's also immune signaling that goes on in the brain. There's some immune cells that can get into the brain. So they're not exactly sure what leads to, like, say, depression, for instance, right? Some other diseases that are not psychologically related, like diabetes or rheumatoid arthritis, there's just an overactivation of the immune cells in the area, or there's a positive feedback where some cells are just emitting these pro-inflammatory mediators without cease, right? And then you get chronic inflammation, you get fibrotic tissue, and you get the wrong sort of tissue in an area, which sort of enhances that. But it's really interesting. A lot of people are investigating the link between inflammation and depression right now, but it's not totally understood how that works.
Physiologically, do we know like what kinds of concentrations these things need to be at to elicit the resolving response or whatever at the inflammation site?
Yeah, there's been some studies on that, and like as far as a practical point of view, they've shown that it's like, it's not as easy as like eating salmon once a week to boost your omega-3s up there, because there's like so much else in the diet that's getting omega-6s. So I mean, one of the unfortunate things that we learned is like, you basically have to eat like salmon, twice a day in order to get your omega-3s up to a really good level where you're going to see clinically relevant changes, which, if in some countries, they do eat that much fish on a regular basis and they don't eat much red meat. But most of us are not in the habit of eating, walnuts and flax seeds and chia and salmon at every meal, right? So I think it does take a little bit more than just taking, one supplement every other day or something like that.
But like in the inflammation site, like, I don't know, we're talking about fatty acids on membrane lipids.
Yeah.
And there's a lot of them in a membrane, and the cell's just picking some of them out to modify to make as signaling molecules. So I just am curious about the concentration of the signaling molecules in vivo? I mean,
Yeah.
it would be hard to measure. I don't know how you'd know, but I think these are signaling molecules, and I assume they're.
Yeah, I think the signaling is at like low micromolar
Yeah,
levels,
yeah.
which is relatively high
That
for,
is high,
for
yeah.
some signaling molecules. Yeah, and so I think you're just.
So you do need a lot.
Yeah, you do need a lot. And I
Yeah.
think the phospholipase is just kind of doing a sort of like a statistical sampling of whatever happens to be nearby in the membrane, right? So I mean, you're still always going to get some degree of omega-6s coming out unless you've got this diet that's way shifted, right?
And how stable are they once they're?
Yeah, there's different sort of stability. Like there's some other things that we didn't talk about a whole lot. Like initially when the lipoxygenase and the cyclooxygenase make them, there's peroxides. You just put the two oxygens in. So you need like some glutathione around to reduce it a little bit to make it more stable. So it depends on like the general oxidative state of the cell then, like whether or not these have like an intracellular or whether they can act in a paracrine fashion. And so like for instance, there was some studies that were looking at this for ischemic stroke, because when you have an ischemic stroke, all your ion gradients get dysregulated. And the lipoxygenase can get activated by calcium. And so when you have an ischemic stroke, you get all these, there's no oxygen at all, but you have disrupted ion gradients. So there's all this calcium around, and the lipoxygenase is like, okay, let's go. And then when you get, when you reperfuse that injury, you get this rush of oxygen. Lipoxygenase, boom, 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 just starts reacting with everything around it and creates this huge explosion of inflammation, right? Because of the oxidative state of the cell that time. So one idea was to use lipoxygenase inhibitors to prevent reperfusion injury and stroke, kind of similar to that. So yeah, and looking at other oxidative diseases where there was high amounts of free radicals and extra oxygen that could activate the lipoxygenase were times that we thought an inhibitor would be good to prevent, the too many random signaling molecules getting made at a time.
Right. Yeah. I have one other sort of off a different tangent question, and so early in your talk, you talked about the cyclooxygenases and them being involved in the inflammatory response. Are those what the COX-2 inhibitors like Vioxx and Celebrex and all that are targeting?
Yeah, there's
COX?
two different cyclooxygenases, and they're called COX1, so COX1 and COX2, and they're and they make different. Yeah, they make slightly different profiles of molecules, and I think COX1 is more important in. Let's see, I kind of forget, because they did Vioxx, which was a COX-2 selective inhibitor, and I think that's because those had more that had more pro-inflammatory effects. So if you shut that off, it was good, but you maintained some of the good effects that could come out of COX-1, right? So it's like some of the research we did was specifically looking at, could we get an inhibitor that would just target 15 lipoxygenase, but not 12, or target 12 and not 5, something like that. Yeah, and there was all the issues with Vioxx because it actually reduced pain, but it actually increased your propensity to clot. So it led to these higher incidence of heart disease actually, and counterintuitively. So then they had to pull it off the market. Yeah.
Celebrex is still around, right?
Is it? Yeah, Vioxx. Yeah, I think Celebrex is around, but Vioxx is not.
No, Vioxx.
Yeah, that's a good question. Yeah, and it's really hard. There's some of the trouble of studying this in a way is that there's lipoxygenases and cyclooxygenases like everywhere, and they're producing all kinds of molecules. So it's really hard to tailor down the effects unless you have a good inhibitor that can focus on one area or one tissue, right? It's not like some things that are expressed in one location and it's really easy to inhibit.
Just going back to talking about the link between inflammation and depression. Earlier in your slide, you briefly mentioned that cortisol can inhibit a certain stage in the process of inflammation. Are the studies that have been linking depression to inflammation, like talked about that specific thing at all? Do they focus on cortisol?
That's a really good question, and I don't know. I would just hypothesize now without having too much to back it up that because cortisol, definitely your stress hormone, which is cortisol, could have a profound effect on your level of stress. And you can see definitely in the cortisol has a daily cycle, right? That I think it increases more like during night or day. But so if you ever get sick, you notice that you feel worse at different times of the day. And a lot of that is due to this cycling of cortisol, right? And if you have baseline really high levels of cortisol, that can change, it's a stress hormone, so it can change your immune response as well. So just being stressed or depressed or being sort of you study mice that are in low social status, right? And they get stressed out all the time. And that can downregulate your immune system, or it can cause all kinds of different problems depending on what else is going on with the mice. Good question.
All right. Well, thanks again, Steve. Thanks for giving the talk for us. Thanks for coming, everybody. All right.